Parthenogenesis in Animals by John Wagner, Domingo Uceda, Paige, Gudim, and Dalton, Sherwood. Parthenogenesis is simply defined as reproduction without fertilization. The name itself comes from the Greek words parthos, meaning virgin, and genesis, meaning creation. There are two main forms, cyclic and complete/facultative. For cyclic parthenogenesis, the sexual generation alternates with the pathogenetic generation. Each cyclic organism has its own unique process that determines how and when each type of egg is laid, and thus what type of organism it will develop into. An example of this are water flies, as illustrated below. Some of their eggs are capable of fertilization, while others are not.
In complete or facultative parthenogenesis, the species has the ability to produce sexually through fertilization or asexually through parthenogenesis. These species can create eggs that are capable of development from either fertilization or parthogenic activation. This switch can sometimes be based on the organism's environment. In apomictic parthenogenesis, the offspring is a genetic clone of the mother. In automictic parthenogenesis, the offspring are homozygous from one allele of the mother.
Platythyria punctata, a genus of ants, is an invertebrate that reproduces through facultative parthenogenesis. The colonies in certain locations have capabilities to reproduce through thalidicy. Thalidicy is an example of parthenogenesis where the females are produced from unfertilized eggs, resulting in males being rare in the population. Parthenogenesis is most prevalent when resources are limited. For example, at higher altitudes, lack of moisture, and in disturbed changing habitats. Also, it is common in island habitats at low sea levels. With a limited space and resources, it is most efficient for the organisms to reproduce asexually to conserve energy and avoid courtship. The females that are able to reproduce through thalidicy are advantageous in these situations. Platythyria punctata is unique in that they have co-occurrence of sexual reproduction and thalidicy. The Caribbean populations were compared with the mainland populations using a technique called microsatellite loci to detect genetic variation. 36% of the colonies in the Caribbean had variation. However, 83% of colonies on the mainland had genetic variation. This demonstrates that sexual reproduction was occurring on mainland, but parthenogenesis was prominent with Caribbean populations. This is known because the genetic variation with asexual reproduction is very low. The mainland also shows a higher genotype to individual ratio and genotype to colony ratio than the Caribbean. The genotype to individual ratio approaches 0 in an ideal clonal population and 1 in a sexual population. The ratios are closer to 0 in the Caribbean island population, and the ratios are closer to 1 in the mainland. This supports that there is more variation, thus sexual reproduction is more common on the mainland than the Caribbean, where thalidicy is the predominant mode of reproduction. The expected and observed heterozygosities are compared for the populations on the Caribbean and mainland. The values of the Caribbean islands were more negative, which reflect the genetic similarity. The populations in the mainland were less negative, showing genetic variation. While parthenogenesis is more apparent in invertebrates, like these ants, this next example is a successful example of parthenogenesis in a jawed vertebrate species.
In December of 2001, the virgin birth of a hammerhead shark was recorded along with the discovery of parthenogenesis in cartilaginous fish. Dr. Damian Chapman and colleagues discovered that one of 3 female hammerhead sharks, who were all taken from the Florida Keys when they were less than 1 year old, had a pup in isolation from males. These females were immature and unable to reproduce when they were taken from their source population. Therefore, the common hypothesis of sperm storage, which is the usual explanation for delayed egg fertilization in hammerhead sharks, was not viable. Dr. Chapman hypothesized that the pup was born through parthenogenesis. So how was this tested and supported? Tissue samples were taken from each candidate mother and the pup. The CM 1 through 3 in the left column stands for candidate mother 1 through 3. With these tissue samples, the same microsatellite marker loci technique as in the previous case was used to genotype each shark along with 119 sharks from the source population. This table shows the alleles at each of the 4 microsatellite loci of the 3 candidate mothers in the pup. First, The actual mother can be determined by simply seeing which candidate mother has at least one allele at each locus that matches the pup's alleles. So, by this process, it is easy to see that CM2 is the actual mother. The other two candidate mothers are ruled out due to mismatches. Next, the possibility of automictic parthenogenesis had to be proven. Proving this includes showing that the pup received all alleles from the mother, which we have already done, and that the pup is homozygous for every allele, which you can see here she is. Therefore, the hypothesis of the pup being born from an unfertilized egg was upheld. This is an example of automictic parthenogenesis because one allele was received from the mother, which was then duplicated. This is why the pup was homozygous at every loci. With this discovery of parthenogenesis in cartilaginous fish, asexual reproduction has now been demonstrated in all major jawed vertebrate lineages, except for mammals.
Advantages of parthenogenesis include superior reproductive potential and colonizing ability, because every individual can potentially found a population, as well as protection against the breakup of favorable gene combinations likely to occur in normal meiosis and when sexual isolation occurs. Disadvantages include any major change in environment can completely wipe out the species due to the lack of genetic diversity, as well as any negative mutation is easily passed and cannot leave the life chain.
Very specific environmental factors can cause an animal to reproduce through parthenogenesis. This change gives the animal an advantage in reproduction ability and a greater fitness in its specific environment. Animals usually do not reproduce through parthenogenesis, however, illustrating the importance of having genetic diversity and the ability to adapt to changes in the environment.